In the last chapter, we covered the first and second lines of defense, as well as introduced some of the various blood cell types. In this chapter, we'll be moving on to look at the third line of defense, that is, specific immunity. So when the barriers and non-specific defenses fail, the immune system has one more trick up its sleeve, one more mechanism to resist infection. The third line of defense, or acquired specific immunity. Acquired specific immunity is the resistance developed after being exposed to a pathogen. It's acquired only after an immunizing event such as an infection or vaccination, and it provides long-term protection against future attacks. And as we'll explore, it's a product of a dual system using both B and T lymphocytes. The ability of our body to react to foreign substances is called immunocompetence, and immunocompetence occurs during development of the B and T lymphocytes. These lymphocytes become specialized for reacting to only one specific antigen, otherwise known as immunogen. Antigens or immunogens are at the center of the third line of defense. An antigen is considered to be any molecule that stimulates a response by T and B cells. Usually it's a protein or a polysaccharide that's found on or in cells and viruses, including those labels on our own cells. However, our immune system generally doesn't attack our own cells. So usually they're only foreign molecules that can stimulate an immune response. But keep in mind that any exposed protein or polysaccharide is a potential antigen. There are two characterizing features of this third line of defense. First of all, it is very specific. So the T and B cells learn to destroy only a specific immunogen. And they'll retain a memory of that immunogen so that if it invades again, the immune reaction is very fast. There are 5 main stages of immunologic development, with lymphocyte development and differentiation. Move on into the presentation of antigens, and then the path of development splits significantly. We're going to see that we're having the challenge of both B and T lymphocytes by the antigens here and here. And then we'll see that the B lymphocyte response and the T lymphocyte response. The B lymphocyte response is humoral immunity, and the T lymphocyte response is a cell-mediated immunity. We can also look at this in a different sort of way where we have stage 1 and stage 2, and then we see a branch where B and T challenge is a little bit different. We're developing different responses, either the antibody or humoral response or cell-mediated response. Throughout the rest of the chapter, we'll look at each of these different stages in detail. But first, a quick overview. So let's begin at the beginning. Lymphocytes are central to the immune responsiveness, and their development, as we've already explored, begins in the yolk sac and then shifts to the liver and bone marrow. Both types of lymphocytes arise from the same basic stem cell type, and then they diverge into their 2 distinct types of T cells and B cells. The final maturation of B cells occurs in the bone marrow, and the final maturation of T cells occurs in the thymus. Once they've become mature, they migrate to separate areas within lymphoid tissue. So they may both go to the lymph nodes or to the spleen, but they'll reside in separate areas of that organ. And it's not that they stay there entirely, because there's constant recirculation through the blood and the lymphatics as they circulate through the system and the lymphoid organs. B cells and T cells work very closely together in the immune responses. There's a high level of communication between the 2 cell types. In fact, one cell type may influence how the other cell type acts. When antigens are presented, both T cells and B cells are activated. When foreign cells enter the body, they've crossed the first line of defense and they enter the tissue. And the phagocytes, as we explored in the last chapter, migrate to that site and start cleaning things up. And then we have some antigen-presenting cells. We've already looked at macrophages and dendritic cells. The macrophages will ingest the pathogen and induce an inflammatory response if it's appropriate, etc. And then the dendritic cells are going to ingest antigens and migrate to the nearest lymphoid organ. that nearest lymphoid organ is probably going to be the lymph nodes that drain that particular area. Here, in these organs, they're going to process and present the antigen to the T lymphocytes. So macrophages have ingested the pathogen, dendritic cells ingest the antigen, and then they take it to the lymph organ so that they can show the T lymphocytes what's going on out of the scene of invasion. Also, pieces of the antigen will drain into the lymph nodes. These pieces are the ones that are going to activate the B cells. When the B and T cells are challenged by an antigen, as in they've been presented with an antigen, they will proliferate and differentiate. This creates a clone, or a group of genetically identical cells. Of these clones, eventually, some of the B cells will become B memory cells, and then some of the T cells will become T memory cells. And these are going to be used to ensure future reactiveness against the antigen. Now, the progeny of the dividing B cells are called plasma cells. These plasma cells are programmed to synthesize and secrete antibodies into tissue fluid and blood. When the antibodies attach to the antigen is now marked for destruction and neutralization. So the antibody is attaching to the antigens. Now these small particles of antigen are marked for destruction. This is the basis of humoral immunity. And again, we'll explore each of these steps in much more detail. This is just an overview. T cells respond in a different way. When activated by the antigen, the T cells give rise to one of three different types of progeny cells. We'll have T helper cells, T regulatory cells, or cytotoxic T cells. The helper cells activate macrophages and help activate the cytotoxic T cells. The regulatory T cells are there to regulate the immune response of the other cells. They're pretty much the self-check system of the immune response. And then cytotoxic T cells are the ones that really do the destruction. They destroy infected host cells as well as other foreign cells. So that was a super quick overview of what happens in each of the stages, just so that we can see the big picture before we move into the details of each stage.